am Dr. Sagar Kumar. By the interest, I am having the interest to educate the people mainly in the need orientation. For that, I think for so many times, so a lot of time, and to start a YouTube channel. My channel name is DK Neat Botany. In this channel, all the botany part, whatever the part comes under the biology in the NCERT for the CBSE class will be covered. In that one, first I chose the plant physiology. In the plant physiology, there are the three topics. One is the photosynthesis in higher plant and respiration in the plants, and third one is the plant growth and development. Out of this, we are starting today the photosynthesis in higher plants, which is largest chapter than other two in the physiology. And their chances are more to test the questions in this chapter also. So, and one more thing, in the NEET, in the previous years, generally he is giving 2 average questions, but don't think like that. Chances are there. There is no rule how many questions should be given from each chapter. But for that one, you must be prepared properly in each and every topic. You must be like a soldier. In any angle also you have to attend the question and you should get the good marks. Now, I am going to do this chapter complete in detail. Concept orientation. Concept orientation is required for the NEET. Means that if you know the concept, then you will give the answer properly. Now you see and listen properly and understand, try to understand the concept and read the textbook. And one more thing is, I am perfectly following the NCERT textbook. And in this chapter, sub-chapters also he has given. Each and every sub-chapter I used to follow, and each in paragraph, whatever the important contents are there, those things I will cover it. So, and in the NEET orientation, which type of the questions will be created also in the chapter I will discuss. Now you see here, for example, the chapter heading is photosynthesis in higher plants. Photo means light is also called as photo because it consists of the photons. And the light is synthesizing. What it is synthesis? That is synthesizing the food material. So the photosynthesis means light energy will be used for the synthesis of the food material, and that is the process is called as photosynthesis. Now that process in higher plants we are going to study. Higher plants means what? In the plant kingdom, higher plants are nothing angiosperms. So, flowering plants. So, in the flowering plants, photosynthesis we are going to see in this chapter. And what is the main object of the photosynthesis? It used to use the light energy and converts it into the food material. So, what is the use of food material? Now you see, food is organic compound. In the photosynthesis, whatever is forming, that is the organic compound. Example is the glucose, C6H12O6, all of you know that one. So, food is organic compound. Of course, when we are eating the food, it is organic compound. Of course, inorganic water and salts will be there. But organic compound is important. So, such type of organic compound, when we take in that one, it gives ATP and forms biomolecules. Now, if you go to the chapter biomolecules, there he has written clearly that is all the carbon compounds present in the living organisms are biomolecules. Go and see that one. If you look at it. Now, such type of the organic compound biomolecules will be formed and gives the ATP. All of you know, all of you are familiar with the ATP. ATP is the energy currency that will be discussed detailedly in the respiration in the plants. Plants are autotrophs. Why? Plants are doing the photosynthesis. Photosynthesis is nothing, it is preparation of the food material. So only the green plants can synthesize their food material for their purpose, living and all the living activities. What are the activities? That activity is nothing. It is growth, reproduction, and metabolism, and all the other things. Now, whenever we are seeing this one, the plants are preparing their own food material through the photosynthesis process. So they are called as autotrophs. Now, whenever food material by the photosynthesis, how they will synthesize? They will synthesize own food material, food by the photosynthesis. Such a type of thing, photosynthesis means preparing of the own food material, is not present in the animals. So they are, the animals are heterotrophs. Why? Because for the food material, they are depend on the plants as the herbivores. This you will know very well in the ecology. Plants are the primary producers and consumers will be the primary, secondary, tertiary. All these things you know in the 10th class, below 10th class also. So it is okay. So as they are depend directly as the herbivores or indirectly as the carnivores, that they are the heterotrophs one we used to call. Now, how the photosynthesis will be done? The photosynthesis will be done in the green plants. In the green plants. The green, why we are telling particularly green is nothing, it is the pigment. That's the chlorophyll that you know in the below 10th class also. So, photosynthesis is done by the green plants. By which process? Through the process is called as physicochemical process. So, what is physicochemical process? Physico means here the sunlight. It is the physical energy which is coming in the nature. And it will be converted into the chemical energy. Means that is the chemical energy is nothing, it is sugars. Sugars which is the food material. So it is preparing the food material through the sunlight. That is the thing. That is called as physicochemical process. Remember it. Because one more is coming. That is photochemical. Light reaction is photochemical. But total photosynthesis we used to call it as physicochemical process. This, whatever the equation I have given here, it is not present in the textbook for paragraph. But for your better understanding, I use this formula which is coming in the next after two subchapters. That here, whenever we are seeing, that is carbon dioxide, that is 6 molecules in the balanced equation. 6 carbon dioxide molecules, 12 water molecules in the presence of the light and chlorophyll in the plant. It will produce C6H12O6 and 6 oxygen and 6H2O means water. This is important. This is also very important, which is the byproduct. But the main object of the photosynthesis is this one. It is the byproduct. Now, so photosynthesis is a this is called as anabolic. Why? Carbon dioxide and water molecule, molecular weight is less, small molecules forming the big molecule. So when small molecules are gathering together and binding and forming the big molecule, that is called as anabolic. In that anabolic, to combine these two, it required energy, that is the light energy. For that one, some base must be there, that is cell organelle in the cell, that is the chlorophyll. Now this will be discussed after one more subchapter. So finally what we want to conclude? that without sunlight energy, this process cannot be done. Sunlight energy only is converting to the food material as a physico-chemical process. So sunlight is basis of life on earth because it is useful food material for not only the plants and on the plants dependent heterotrophs also. So overall for the life, the basis of the life means all the living organisms used to depend on this one. Now here it is a vital product also. Food is most important. Without that life won't be there. And out of first vital product is food, that is the C6H12O6. So in this one glucose will be first prepared. Okay, that we will discuss later on, no problem. Now the thing is, one more vital product is also coming through the photosynthesis. That is important. That is the oxygen. And why the vital product oxygen? Oxygen is very important for the respiration, aerobic respiration. Without oxygen, aerobic respiration won't be there. Majority of the land, living organisms which are on the land, which are on the earth, they do the aerobic respiration because the food, for complete energy of the food material will come from the food material will come through the aerobic respiration that we will discuss. So sunlight is the basis of life on earth because sunlight means it is nothing, it is energy. That energy is converting by physico-chemical process and giving us food. Not only food, one more vital thing which required for all living aerobic respirative animals, aerobic respirative organisms. Oxygen is also coming from this. So photosynthesis is giving two main vital things. One is direct vital thing, it is done by the food material, and as a byproduct it is giving the oxygen. So he had asked one question. If the photosynthesis is not producing the oxygen, what is the condition on the earth? On the earth, only anaerobic living organisms can survive. Because as it is giving this much, we are surviving. We are also aerobic. That we will discuss detailedly respiration in our respiration in plants. Now, one more sub-chapter he has given. He has given the sub-chapter numbers. On that one, what do we know? What do we know? Means here, if you are in the 11th class chapter it is. So, before 11th class, what you learn about the photosynthesis through the experiments that he has given here. That is, through three experiments he has given. These three experiments are given in the 10th class and the 9th class in the CBSE. That is the NCERT book. First one is variegated leaf experiment. For what the purpose of this experiment? Chlorophyll is needed. Chlorophyll is needed for the photosynthesis in presence of the light. So chlorophyll, where it is to present? In the green parts. Means the chlorophyll is there, then it will be in the green color. So variegated leaf means the croton leaves whenever we are seeing, they are in the multicolor. And sometimes it used to have like this also, green and white. White patches also used to present. Such type of the plant leaf we have to take. And if the plant is in the sunlight, and remove this leaf and do it the test. The test he has given. Test is nothing, iodine test. Either when iodine test you are doing, where the starch is there, the iodine will convert into the blue color. That is a blue color. Here, the leaf itself, it is having green and white or other color. Wherever the green parts are there, that only will be converted to the blue color, because that part only preparing the food material sugars, which will be converting into the starch. Iodine test used to say that where the starch is there. In the photosynthesis, starch will be prepared to the product. So like this, we can observe. And here, the green parts, whatever are there, that only is converting the blue color. It indicates it is preparing the food material in the green color. That is the chlorophyll is required. Next one is light is needed. Light means here, the light is required. It can be proved by the plant. By a plant, that in that plant you select a leaf and cover it with black paper and keep this experiment few hours in the sunlight, and then later on you remove this leaf and test it. That is the iodine test. Then in the iodine test you will get the positive result, means blue color changing. It will be the part of the plant which is not covered will be converted into the blue color, because that is the green color, the entire leaf, but the part is covered by the black paper, so that part is not converted. So in that part, what it is losing? Light it is not getting. So light it is not getting, it is not preparing the starch. This is the second, that is leaf partly covered experiment. And third experiment, that is off leaf experiment by more. What the object of the experiment? Now the carbon dioxide is required for this photosynthesis. That can be proved by one more experiment. In this one, you have to take a plant, potted plant, and select a leaf, cover the leaf. That leaf will be kept in a test tube. And by in the test tube, before keeping the leaf, you have to keep the potassium hydroxide liquid soaken that cotton. A cotton piece you take and keep dip in the potassium hydroxide and keep that one and set it up for test tube. You have to keep cover the half of the leaf, some of the leaf, half of the leaf you should keep. You can keep the test tube, and you have to close it carefully, that without damage of the leaf, and keep this experiment in the sunlight for few hours. Later on, you remove this leaf and test it. That is the iodine test. Whatever the part is present inside the test tube is not converted into the blue color, and the other part which is outside the test tube converted into blue color. Now the question comes, what is the part which is present in the test tube is not having? The main thing is potassium hydroxide, which is present in the enclosed leaf in the test tube. Is in that whatever in this one, whatever the air is there, in that air carbon dioxide is absorbed by the potassium hydroxide. So that air whatever is present in the test tube is not having the potassium hydroxide. So that is not having the carbon dioxide. So here carbon dioxide is removed. So without carbon dioxide it is not possible to prepare the food. So all these three experiments. So what is the use? Now you can say one thing, don't underestimate these experiments. It can become in the matching. It may come multi-statement, how many statements are correct. Anything can be asked out of three experiments, multiple choice questions. So in this way, these three experiments concept you should know. That is the thing. Now we will see the early experiments, one more subchapter. Early experiments means up to the 1770, how the plants are useful for the human survival. Means they they are using, but by which mechanism, how it is, that they don't know. But for that one, when they are trying, early experiment shows stepwise development. That first step, that is 1770, Joseph Priestley done the bell jar experiment. In the bell jar experiment, here, what is this bell jar experiment? What is his observation findings? Of course, some people are teaching this one in the wrong way. The main object is, this bell jar experiment is meant for the air. Air, outside air, it is, we are the air, which are of the air is spoiling by the living organisms. Now for that one, take a bell jar, and before bell jar you have to cover it on a burning candle, and keep a mouse also, you kept a mouse also in that one. When this bell jar inside, the air is completely, it is not having connection outside air. So this air, it is having the no connection outside air. So in this completely closed chamber, whatever the air is there, that air will be consumed by the burning candle and the mouse. What happened? After few minutes, 1 or 2 minutes or something, the light, whatever the candle is extinguished, means it stopped burning, and the rat, whatever mouse is there, it is suffocated and died. So he thought, something it is doing. These both are present in the closed chamber, air. They are doing something. And then he introduced the same experiment he conducted and kept a potted plant, that is mint plant, in the bell jar. Then all are mouses are alive in condition, and the candle is in the burning condition. Nothing happened to the plant also. So all these things he has not done simply. A series of experiments he conducted. And remember one thing he has given in the textbook. Joseph Priestley discovered the oxygen in 1774. By the 1770 he don't know the oxygen. So in his conclusion he told that whatever both the living mouse and burning candle has damaged the air by taking some part of the air, and finally they extinguished and died. But when he insert a potted plant in that one, all are in the same stage, means mouse is in the living condition, candle is in the burning condition, nothing happened to the plant. So he concluded that whatever the damaged air is damaged by the mouse and the burning candle are restored by the plant. So what is the Priestley experiment, bell jar experiment? That is these are the things you should remember. Next, one more experiment by the John Ingenhouse. This is also most important. John Ingenhouse experiment. He showed 3 conclusions that is important. Some people are ignoring. What he done, textbook also has written clearly. Read it properly. That is, sunlight is required. Means in the 1770 only, when he done this experiment, he published. By reading that one, John Ingenhouse conducted the same experiment. Whatever Ingenhouse that Priestley has conducted. But he done some additional thing. What is that? He kept the same experiment in the sunlight and same after without sunlight. Without sunlight, that whatever the air is damaged, In the sunlight, all three are like this. He concluded that sunlight is required to restore the air, whatever used by mouse and burning candle. That is the first observation, first conclusion, remember it. And he didn't told in that one oxygen because he had conducted this experiment 1770. At that time they don't know because 1774 only oxygen is discovered by the Priestley. Next he extended this experiment in different way. That is, he has taken he conducted one more experiment. That is, he taken the aquatic plant and kept it in the water and he kept a test tube on that one. And the set of the experiment he kept in the sunlight and then he observed some bubbles are coming out from produced by the aquatic plant. So bubbles are coming in the presence of the sunlight only. Without sunlight it is not coming. So the bubbles are coming in the sunlight, something is happening in the sunlight, bubbles are coming. And later on, this is also Indian House also conducted series of experiments. One experiment like this, then this like this, and finally one more thing. By that time, 1774 came. Indian House, that whatever the Priestley has discovered the oxygen, that whatever the bubbles are produced by this one, he observed and he tested and found that the oxygen is coming from the plants in the presence of the air. That is this one. Now here the thing is, here he observed 3 points. Sunlight is required and green plants will produce the bubbles, and from the green parts only, that also important. Green parts only will produce the bubbles which are oxygen, which he concluded later. All the 3 points are most important. He may confuse if the question comes in the NEET. So these are the sunlight experiment, that is actual. And green parts will produce aquatic and bubbles. And those bubbles produce gas is nothing, oxygen. Later on he discovered after 1774, because after 1774 only that Priestley has discovered that. Now one more experiment, and these 2 experiments are important for the production. Something is restoring, and restoring is nothing, it is oxygen. Up to there, up to 1774, up to that one. Now 1831, we gave his sketches. He is a physiologist. He done some experiment, series of experiments, and found, means proved, that is, plants produce glucose when they are growing. Plants, when they are growing, they will produce the glucose, and that glucose will be converted into the starch. That is proved. And next, in continuation of the experiments, he found that where the glucose is producing, the glucose is produced in the green bodies of the leaf. Means green, not only leaf, any pair, green part is there in the plant, there it is producing. So the green bodies are nothing nowadays, we know, even you also, that is the chloroplast. And the chloroplast contains the chlorophyll. That is the thing. So chlorophyll used to prepare the food material. Who has proved, means this fellow, Indian House, that is the sketches. Who told that the oxygen will be produced in the green parts, Indian House. And plants used to produce that whatever used in the part of the air, plants will produce that part and restore. Who told? Xanthophylls. Questions will come like this. For that one you should have some concept. This concept is, but the green parts and sunlight is required, and this part is food material will be prepared by the plant. Just this. Now, one more experiment conducted by Engelmann. That Engelmann, what he has done, he done the experiment by splitted light. That is the prism. Whenever you pass the light through the prism, it will be splitted into the 7 colors. All of you know that the prism, that is the spectral components. All the 7 colors will come. This is the prism. Light is coming and it is splitted. That is the colors are nothing. VGR and violet, indigo, blue, green, yellow, orange, red. That is visible, like in the visible light. So he want to know which color is most important for the photosynthesis. That he want to observe. For that he has conducted, he taken this one and kept water and taken cladophora, the algae. Algae cladophora, how it is? Like Ulothrix, you have seen in the algae that chlorophyll, that is an algae we studied. It is a threaded like thing. It is like this. Cladophora he has taken. On the cladophora, before keeping in the water, he done, he suspended, means he has given, means he added some aerobic bacteria. Aerobic bacteria means which required oxygen for their survival. He also added some aerobic bacteria in that media. Now after few hours, he observed the prism components, whatever are there, that whatever the light colors are there, he observed that blue and red light places the bacteria are gathered more. Means what is this? But they are the aerobic bacteria. Where the oxygen is available, there they will develop or they may go. So blue and red light places only the aerobic bacteria concentration is very high. Of course, it is present other places also very few, but maximum concentration gathering is at the blue and red light. And he told blue and red lights are important for the photosynthesis. Why he used bacteria? Because photosynthesis will, when the photosynthesis is going on, oxygen will come out. So oxygen is required for the bacteria as the aerobic. So where the oxygen is available, the bacteria will grow. For the indication where the oxygen is coming, means the photosynthesis is going on. That is the thing. For that only we have done this experiment and concluded that is the blue and red lights are important. The same thing will come after one more sub-chapter. That is the absorption spectrum, and the next is the action spectrum. This is the nothing action spectrum. What is the action spectrum means? That in the how it is, that detail I will discuss there. But I should not go from directly go from here to there. So first action spectrum was observed by the Engelmann. This is about this. Already we came to know that blue and red light are important. Most of the photosynthesis will be done in that one through the experiment of Engelmann. So in the middle of the 19th century, when they reached, the scientists came to one conclusion, that is carbon dioxide, that they given empirical formula, means just they imagine the formula, that is the carbon dioxide plus water in the presence of light, it is to give the sugar and oxygen. What is there? CH2O means it is a part of sugar. So 6 molecules, if you add such a thing, it will become C6H12O6. Okay, it's all right. So what is there? It is wrong, I am telling. You also will say, why? Because this equation indicates that is the carbon dioxide and H2O Here H2O carbon is adding here. That is the thing. This one indicates. Means what? The oxygen is coming from the carbon dioxide. That is proved. It is wrong later on. But that oxygen is coming because in the experiment, early experiment we saw that the Engelmann experiment, that the Indian house experiment, all the things. Now you see here, carbon dioxide, oxygen is coming, sugar is preparing. Equation is like this. They thought, that is, the only thing is, they thought that carbon dioxide, carbon is going and adding to the H2O. Here if you put the carbon, see, it will become sugar. Oxygen is coming out. That much only they given. But it was wrong. That is proved by the Niel experiment. Now what is the CV Niel experiment? CV Niel experiment is called as milestone in the photosynthesis. It is the most important experiment done by the milestone of the experiment in the photosynthesis is when Niel experiment, that is in the 1831. What he done? He done the experiment. He is a microbiologist and he done the experiment on purple and green photosynthetic sulfur bacteria. Photosynthetic means it is using the light. So purple and green photosynthetic sulfur bacteria. Okay, he done. So what he observed, that equation he has given in that one how the photosynthesis will be taken. He had given that your textbook like this 2H2A plus carbon dioxide in the presence of the light will give the H2A plus part of the sugar like this, and H2O water is generating. It is not here. So this is most important. Now the thing is, he is telling H2A is oxidizable. It will can be oxidized. Oxidization means it will liberate the oxygen. And oxygen will come out and it will give the hydrogen. Hydrogen is coming out, that is oxidation. Oxidizable substance plus carbon dioxide gives the sugar and water. That is, in this one, hydrogen, that whatever the hydrogen is present here, hydrogen reduces the carbon dioxide and produces the sugar. So here what we thought, the carbon is coming here and oxygen is coming out. But here he proved that is the oxidizable substance. Here H2A is separated, oxidized and come out and given out the hydrogen. That hydrogen is added in between carbon dioxide. So here then the whatever additional oxygen is here, one molecule is it will be added with that what how many here, 2 only, but how many are there? 4 hydrogens are here. So 2 hydrogens added to this one and oxygen came from here and formed the H2O. Here the H2O is there, no, but H2O is coming. This is the find. Who told in the photosynthesis water will be produced? One Niel. This is the neat question. Remember it. And next, why the hydrogen he has given told, that is the oxidizable compound. Here the thing is, the hydrogen, and after knowing this one, he told, he inferred, means he applied the same formula for these are the sulfur bacteria. So he applied the same thing to the green plants also. In the green plants, what they told, CO2 plus H2O will give this one oxygen. But without water also, water is coming out. That also he had added in this one. That what he done, 6 CO2, 12 molecules of H2O in the presence of the light will give 1 molecule of sugar, that is the glucose, and next will be the 6 molecules of oxygen and 6 molecules of the water. What is this? I can say one thing. This is not explained by many people. Why? Because he asked one question. Why it is taking 12 H2O and why it is giving 6 H2O out? It can take 6 H2O like this and it can produce. For example, you do it 6 CO2, 6 H2O, then it will become C6H12O6 and oxygen. 6 oxygen will come out. But what is the difference between this and that? Here there is no water production. Now come to the point, one important point which we are missing. With even I can say many lectures also missing. That is, now if you see one thing here, it is, now I am writing, you see, here 6 H2A plus 6 CO2 gives, this is, if it is sulfur bacteria, it will become sulfur. This is if it is H2S, the H2A is the sulfur bacteria, it is sulfur, H2S, sulfur dioxide or the sulfate. When at that time it becomes that one, that whatever the A is sulfur, sulfur is coming out. And if it is H2O, what is coming out? That oxygen is coming out from this one, water. That you want to tell. And now you see H2A means 6 H2A plus 6 CO2 gives 6A plus C6H12O6 means one sugar molecule and 6H2O. Same. Here you have to observe why the 12 it has taken, why it has given. Again means here, that is, here it is 2H2A, 6 molecules here 12 it is actually. 12 H2A, 12 H2A means here what? Here it is, now you see CO2, 6 CO2, 12 H2O. Now what is happening? Just you see here, I will write somewhere. That is, for example, C6H12O6. Out of this one, carbon and oxygen are coming from the carbon dioxide and hydrogen is coming from the water. But in this equation, water is getting carbon. That is the difference. Now whenever we are seeing this one, same thing. That carbon dioxide is 6 CO2 means how many carbons are there, oxygens are there? 6 plus 2, 12. 12 carbons are here. And how many hydrogens are here? 2 into 12, 24 are there. So 6 carbons has gone here. Okay. And 6 oxygens only are gone from the carbon dioxide. 6 extra oxygens are there. Remember it. And that 6 extra or whatever the oxygens are there, and 12 hydrogen and 12 oxygen atoms. It is, remember it. It is atoms. How many molecules it will become? Means when the 2 atoms will combine together, it will become one atom, one molecule. So 6O2, okay. This is alright. The oxygen 12 atoms will become 6O2. Good. Then 6 oxygen extra it is having here. The 6 atom, that whatever the 6 carbon oxygen extra, that means carbon and oxygen, here it is, and extra oxygen, and here 12 H2O, 12 are used here, and extra 20 out of 24, 12 are used here, and 12 are extra are there. 12 extra means that 12 extra means 6 into 2, 12. Out of 24, 12 has gone here, and 12 hydrogens. 12 hydrogens means here 6 H2O hydrogens and oxygen 6, which came from this one. What it indicates? It is very important thing. You have to remember it. Many people are not explaining this. For formation of the water, oxygen is coming from the carbon dioxide. That 6 oxygen. And formation of the water, 12 hydrogens are coming from the water. So it is created water, remember it. This is the formed by the metabolism, because here what are the Niel experiment when you are seeing, here the water is produced. Now one Niel experiment we are telling milestone. Why? Because here in the 19th century, middle of the 19th century, they given one formula, that is empirical formula. In that one, carbon dioxide or oxygen is coming. But this fellow proved that is not coming from the carbon dioxide, it is coming from the water. How he proved? And he has inferred, he done the experiment on the photosynthetic bacteria, that is sulfur photosynthetic bacteria. Here, if it is the A is the sulfur dioxide or something, it will come either if it is a sulfur dioxide, that is the hydrogen will go here and sulfur will come out, sulfur is coming out. And if it is the hydrogen sulfate, sulfate will come out. That only in the textbook he has written either sulfur or sulfate will come. That is the meaning, remember it. So, from the H2A, if it is a sulfur dioxide, sulfur will come out. If it is sulfate, hydrogen sulfate, sulfate will come out. So, that hydrogen is coming from the oxidizable, means that is the H2S or H2SO4, HSO4. That hydrogen is coming from this one. That hydrogen used to reduce the carbon dioxide and forms C6H12O6 means CH2O, part of sugar, that you should remember. And here water is not there, but water is formed. This is the important. Even though water is not there, water is formed. Same thing inferred for the green plants and told 6 carbon dioxide plus 12 H2O in the presence of the light will give C6H12O6, that is the glucose, 6 molecules of the oxygen and 6 molecules of the water. And he has given in the textbook, remember, why it is taking 12, why it is giving 6. If you take 6 also, if it is take 6, it will become this. In the 19th century, whatever they given, empirical formula. But here, he proved that water is formed water it is. That is metabolic water, we used to call. One more place also metabolic water will form in the respiration, that I will tell there. It is, when he asked this one, what you want to know, that is, this is carbon dioxide used to provide carbon and oxygen for the, this sugar, this and this both are coming from the carbon dioxide. And how many, so, but it is having 12 oxygen here, but 6 oxygen only coming, 6 carbons, okay. Now, here 12 H2O he has taken. Out of the 12 H2O, how many hydrogens are there? 24 are there. Out of 24, 12 are used here because it came here according to this formula. That is H2A hydrogen oxidizable. Now, if you see here, the H2O 12 are used. 12 extra hydrogens are here and 6 oxygens are here. So 6 oxygen and 6 into 2, 12 hydrogen united and form 6 molecules of the water. It is called a generated water, not from this water directly. For the formation, he will ask how the water will form. Important question for the NEET, remember it. Water is generated water by taking oxygen from the carbon dioxide and by taking the hydrogen from the water. Then only it is what this is also called as metabolic water. And it also proved by the using of radioisotopes, that is O18 in the water. In the water, hydrogen, oxygen, whatever is there, they given O18 form and they given it to the plant. And that H2, whatever it is formed, oxygen, that is O18 water, O hydrogen, that is the radioactive oxygen came here. It indicates the water is coming, the water is giving the oxygen. That also he has written in our book. And one more question he has given in your textbook, that is, why 12 molecules it is there, means it is 12 molecules H2O water. But here the carbon dioxide are participating in the formation of the glucose, carbon dioxide. So 6 oxygen are more, that 6 oxygen here, and here 12 out of this 12, 24 hydrogens are there, 12 are used here according to this one, and next 12 are extra there, that 12 came here, 6 into 2. So it is a generated water which is called as a metabolic water. Now this it indicates here in this one 19th century whatever they given, oxygen is coming from this one because carbon came and sit there. But here he proved that the oxygen is coming from the water, not from the CO2. Who proved this? This he will ask definitely. That is the Niel experiment. And who this is inferred. But later on some scientists proved it by giving the radioisotope, that is the O18 in the H2O. That O18 came here. Means that oxygen whatever is coming, not from the carbon dioxide, from the water only. This is the important thing, and it is called as milestone experiment. Remember it. And you tell your friends also to solve this one. That I am very much confident why it is, how it is. Many people are trying to escape it, no doubt, I can say. But I am not telling you, I am not objecting that person. But you have to think little bit more. All are competent, no doubt. But the explanation is this one, remember it. Tell your friends also. Now, this is the one Niel experiment. Why it is called a milestone? That also you can ask. What he told, that oxygen is coming from the water only, not from the carbon dioxide. Proved by, he will ask. How many questions can be generated here? Please remember it. Now, you see here, one more subchapter he has given. Where does photosynthesis take place? Where it will take place? In the chloroplast. That chloroplast structure, you know, in the cell organ, in the cytology. That cell contains of the different cell organelles. One of the cell organelle is the chloroplast, and its structure given detailedly. That is it will have the 2 layers, wall and outer layer and inner layer. Inside that one, whatever fluid is there, that is called as stroma. In the stroma, membrane system is present, membranes are there. Those membranes is to form the grana, and next the grana will be connected with one tubular structure that is called as stroma lamellae, and all the things you know. But the thing is here, the thing is where the chloroplast is to present. If you start, it is enough, because the detailed structure of the chloroplast is given in the cell structure, cell form, this thing. So they already you know, you have to read there. Now, where the chloroplast will be high, number will be where it is to present. In the mesophyll cells of the leaves in the green plants. Mesophyll cells only will have the chloroplast. No, that question also asked in your book, class book. Any place, yes, wherever the green part is there, in that green part, where it is, the sepals are green, young fruits are green, and whatever the other modification, green modifications are there, that also will have the chloroplast. That is the answer there. So, any green part in the plant, will have the chloroplast, but maximum will be there in the leaves, that is the mesophyll cells. So he asked one more question, that is align. Align means arrange, means how they will be aligned. They will be particular place. Align, this also some people are escaping. That I observed from some people. That is, align means why they have to align, arrange in the properly compared to surface area. Surface area, how they will align in the high sunlight and low sunlight. That we are discussing here, most important, remember it. The thing is, why they have to? Because if the sunlight is very high, that photo-oxidation will be taken place in the chloroplast, which is a damages to the chloroplast. Photo-oxidation will be taken place. Photo-oxidation means newly oxygen won't come directly, won't be come out in the, that way we will discuss later on. In the atom level it will come, later on it will become, immediately it will become O2. Then it will be, otherwise in the atom level it will have the more minus charge and it will be highly reactive also. At that time it will damage the chloroplast molecules. To avoid that one, the chloroplast will be, it themselves will be aligned, depend upon the light to get the optimum light. Whatever required light. So how they will be on the surface area, he has given the question, remember it. When, what is the shape of the chloroplast, you should know. Chloroplasts are present in the oval shape or in the lens shape. So generally majority will have the lens shape. How many is to present in each cell? In the cell means general mesophyll cell, 20 to 40 is to present. To get maximum optimum light, they will be exposed. They will arrange in particular way, depend upon the light. When the high sunlight is there, so here high sunlight is there, if it is the lens, it is the thing, like lens. So if it is like this, it will get maximum light on the surface. But when it is in the light like this, vertical, light will fall less. So that only it is doing nothing. Here it is perpendicular to the surface area. They will align perpendicular to the surface area when high sunlight is there. Why? To avoid the photo-oxidation. These all are important for it. Why? Why they have to align? How do we tell why? All the things are important. Remember, each and every sentence is neat only. If you give that like that question, he will say, my dear student, it is difficult. No, nothing difficult. You have to read in this way. So they will align that perpendicular to the surface area, perpendicular. And when the light is, low light is there, now it is perpendicular. It will be like this. Means that is the, it will be the parallel. Surface parallel it is. Means here the surface will be more, it will get optimum light. That is the thing. So low light parallel to the surface area in the cell, mesophyll cell, and high light it will be perpendicular. Remember it, it is important, definitely you will learn that because this question raised in the textbook, remember it. Now the thing is, in the chloroplast, whenever we are seeing membrane system is, membranes are there, membrane bonded things are present which are called as grana. Grana kinds of thylakoids that you know in that cytology. And grana will be connected by the stroma lamellae. And whatever the fluid is present inside the mitochondria, this chloroplast is called as stroma. Now you remember, stroma is there, grana is there, stroma lamella is there. How they will work? What is there? Where the photosynthesis take place? How it will be taken place? This is kept the thing. In this one, you have to listen. You have to think about it. One thing. Light. Which one will absorb the light? Okay. Which will prepare the sugar? Both the things are separately will be done. That is called as division of labor. In the division of labor, what is the division? The membrane system will tap the sunlight because chlorophyll is to present in the thylakoid, that is grana, and stromal lamellae will have the chlorophyll. It is most important question for the NEET. So they will do the light reaction. Light reaction is also called as light reaction, photochemical reaction. Remember it, physicochemical reaction, there we told it is a photochemical reaction. Light reaction means photochemical. Only light will be taken and some chemicals will be synthesized. What are they? They are the ATP and NADPH. They are the intermediate compounds because they won't be like this. This will be used in the dark reaction. So the products of the light reaction which will be taken place in the thylakoid, that those are the ATP and NADPH will be produced in the light reaction in the thylakoid, which is the membrane system in the chloroplast, and stromal lamella also will synthesize only ATP. This will be discussed detailedly in the C4 plants, remember it. Now, dark reaction and light reaction is separated. In the light reaction, light will be absorbed and the chemicals will be formed, which are the ATP and NADPH. NADP is formed. Now, the thing is, whatever is the carbon, NADP and NADPH are produced in the light reaction. What are the products of light reaction? The NEET question it is. And how it is formed? By taking the light in the presence of the chlorophyll. Now, dark reaction, chlorophyll is not necessary, light is not necessary. Oh, they will depend upon the ATP and NADP, and carbon will be fixed. Means carbon will be reduced by taking off the hydrogen, and that is called as carbon reactions in stroma. So, carbon reactions won't require light and chlorophyll. So, without chlorophyll and light also it will be done. But the thing is, they require ATP and NADPH. If they are not there, this cannot be. Because NADPH, hydrogen, here we told hydrogen is oxidizable. This hydrogen will come and join there, otherwise carbon, there is no question of formation of the sugar. That's all. And these are the division of the labor we can see. And here in this one, you can say alignment is important, how it is. And what is light reaction, what is the dark reaction. Light reaction is also called as photochemical reaction. Dark reaction is called as carbon reactions. And sugar will be prepared where? Dark reaction. Whatever the products required for the dark reaction will be produced by ELAS, that is the light reaction. Light and this whatever the chlorophyll are not required for what? Which one? Carbon reactions. All these are the questions in the NEET. But if a person printed this all the things he will read. This thing. But the concept is, this is the concept. Concept oriented, you have to read, that is I am telling. Now, in this way, these are the things where the photosynthesis take place. Now we will see in the photosynthesis how many types of pigments are involved in the photosynthesis. Means in the pigments also so many types are there. It indicates how you will, physically we can say all the leaves are not in same dark color, light color, pink color, something there. Why it is appearing like that? Because they are containing of so many different types of the color pigments. How we will know? For that one, we have to do the experiment, that is the chromatography experiment, paper chromatography. In the paper chromatography, when you are doing that in the 11th and 12th class experiments, it is there that you know well there. In the through the pigments separated by the paper chromatography is the practical which is present in the CBSE 11th and 12th. So here whenever they are doing that one, here what they will do, I will show you. They will take one small beaker and they will put a paper in the solution solvent here. So the paper they will keep here, that is the chromatographic paper which is having the pores, so because of that one the solvent will be absorbed. Before keeping the paper, here we will put the whatever we crushed, the yellow part, green part, that spot we will keep there. Now the solvent will move in this direction. So whenever it is moving the upwards, this is ascending chromatography, that is not necessary to you. So upwards, that whatever the spot we kept, this whatever the extract that of the green part, that will be separated according to the molecule weight, density. High density will present here, next high density will be here, next high density will be here, low density will be here. Like that they will be separated. When they are separated like this, and different colors we can observe on the chromatographic paper. What are they? They are different colors. What are they? Chlorophyll A will be bright or blue-green color. Chlorophyll B will show yellow-green color. Xanthophylls show yellow color. Carotenoids show yellow to yellow orange. Hello? Remember it? This is asked in the last year NEET exam as a matching. Very important. For that only I am telling you which question he will ask, how he will ask, that we can say, that you have to simply read and the conceptual orientation. This is has given this question in the 2025. So he has given the matching. So they will be separated on what base? Their density base, that you should understand. And in this one, chlorophyll B, which was the highest one, that is not in the textbook, not necessary also. But for your information, in the practicals also, that more high density will be the chlorophyll B because it used to have the one oxygen more, two hydrogen less than the chlorophyll A. So chlorophyll B will be at the bottom, next will be the chlorophyll A, next will be the xanthophylls, and the top and carotenoids to present. Okay, let it be. Now these things are important, how they will be separated. Now this what pigments involved in the photosynthesis. Now we will see the pigments will absorb different wavelengths of the light. So different pigments will absorb different quantity of the light. Different pigments will absorb different quantity of the light at different colors. Means this is the 4 to 500. It is the VGR, violet, indigo, blue used to present in between here. What is this? This entire together is called as blue color. The entire, this will start from the 380. From 380 visible light will start and up to 700 wavelength only the chlorophyll will use. The different pigments will use the light from the 380 to 700. Okay. Now at what wavelength which one is absorbing? The blue color is chlorophyll A and black color is chlorophyll B, and this is the carotenoids. Carotenoids. So different colors I shown here in the graph and the textbook also is shown. So here the thing is, this is the blue color range, and from 60 to 700 red, 750 up to here, but it won't be useful. So 660 to 700 only they will do. And that absorption will be more. 6 from here. Now that we can see, he has given 3 graphs. First graph is light absorption spectrum. Light absorbed, maximum light will be absorbed by the chlorophyll A at in this range. Chlorophyll B in the blue light in this one. Means he has not given particularly, it is not necessary. If it is around 440, it will be the 480 like that, 460 like that. So the peak light absorption of the chlorophyll A will be around this place, B will be in this place. And this is the carotenoids. Whenever you are seeing, here they are starting this one, and they will take some more light. After 500 green will come, some green light also will be absorbed. This is the neat question, remember it. Green light will be absorbed more than the chlorophyll A and B. By, he will ask, that is the carotenoid. Answer. Now, the chlorophyll A and B, when they are traveling through, when they are receiving the light, that is the green, yellow, orange and red. So, here 3 are there. That is the VGR, violet, indigo, blue. This next green and yellow and orange. These are the 3. One is the red. So 7 colors. Out of the 7 colors, whenever we are seeing here, from green to the orange color, the absorption is very less in the chlorophyll A and B. That is the important. That also you can ask in the NEET. And so chlorophyll A and B absorb very less here. Okay. And peak level of the absorption of the blue light is reported from the B, because peak level it is. And in the blue light, absorption of the peak level will be more will be absorbed by the chlorophyll B. And A is the next position. Then the carotenoid is the next position. Because he will ask in the NEET this. How? This is the starting 380. At the 380, the absorption spectrum is starting. In the starting, which pigment will take more, will absorb more? He will ask, tell me out of 3. This is the important question, remember it. Which one? This is the low level, it is middle, this one. This is the forward, he will give question. Chlorophyll A will take more light at the starting point, followed by the xanthophyll, that is the carotenoids, followed by the chlorophyll B. But however, which will absorb more light? In the blue light means chlorophyll B. But in the starting it is less. This is the concept orientation, remember it. Next, even and now where they will be taken less, from the 500 to 650, up to the red light, it is taking very less. When in the red light, that is now we are seeing from here to here, chlorophyll B will absorb less light, but chlorophyll A will take more light. Chlorophyll A light is more, it absorbs more light than the chlorophyll B in the red light. Now the question will rise, that is, chlorophyll A will absorb more in the blue light also and in the red light also. But chlorophyll B will take more. Now you see, from here to here whatever the difference is there, from here to here whatever the difference, it is more difference. So chlorophyll A is the dominant pigment. Now one more thing you have to observe, which is important for the need, that is that carotenoids are taking and they disappeared in the green itself. So carotenoids are not absorbing the light from which one? In the red light they won't. Red light there is no carotenoids absorption because of the wavelength it is not suitable for them. So from the after green what will be? Yellow, orange, red, not at all useful. But chlorophyll A, B, it will run. So in all the lights which will take and which won't take, like that he can ask the question. Now, one thing, and one more thing is here, chlorophyll A and this thing, whatever we are saying, Xanthophyll, that what is the carotenoid contents of the carotenes and xanthophylls, they will absorb in the only in the blue light and some part of the green light. Now you see the action spectrum I am discussing, that how they are in the starting, more will be taken and how it will be taken, these are all the neat questions. So in the starting in 6A, for 380, absorption will be more by the chlorophyll A, next will be the carotenoids, next will be the chlorophyll A. But maximum absorption will be done by the chlorophyll B. And the next, whenever we are seeing in this one, chlorophyll A and B will take less light in the between. That 500 to the up to the 640, then up to 640, 650, where the red light will start. 640 only, 640. So here, there again they will absorb more light in that one also. In the red light, chlorophyll B will absorb less, but it will be absorbed more in the B. It is a neat question. Chlorophyll A is the second position and the difference is less, but here the difference is more. So chlorophyll A is dominant pigment. This he has given in the textbook, also go and see. Now he can ask, the carotenoids cannot absorb after green. That is the important, remember it, shown in this one. This is also a neat question. And next, whenever we are seeing action spectrum. Action spectrum means output out of this one. What came out? Means here, this is the 400, 5 like this. It's in the textbook also. Here, the thing blue light, that is 500, the blue light, maximum photosynthesis has taken place and red light less. Red light it is the maximum it is taken. But in between it is less. This is the action spectrum, what Engelmann showed. That action spectrum, blue and red lights are important, where the photosynthesis will be taken more regard. That is the thing, action spectrum. That there I told, it will come later on, means now this is. Now the action spectrum is how they measure based on the oxygen release. This is most important for the need, remember it. Not direct photosynthesis, how much starch came, how the sugar came, not based on that one, indirectly estimated. What on the based on the releasing of the oxygen. This is important for the need, remember it. Next, one more he has given, action spectrum superposed on a absorption spectrum. What is this? Action spectrums, this is the action spectrum. Superposed chlorophyll is what the relation is. Try to understand it. Now I will tell the answer. Now the thing is, where the chlorophyll A is absorbing more, there only photosynthetic rate is more. Because here, otherwise it must be more here. But it is chlorophyll A where it is present, you see, observe it in the textbook also. So chlorophyll A where the absorption is more, there only the photosynthetic rate is also more. And here it might be here, but chlorophyll A is here, so there only it is producing. For that only we are calling chlorophyll A is chief pigment. So chlorophyll A, wherever is there, there only rate of the photosynthesis is more. So chlorophyll A is called as the chief pigment of the photosynthesis. Now I am telling one more information. I am telling without chlorophyll A there is no photosynthesis. Remember it. And because this question will be definitely will ask in the any exams also. Chlorophyll A is the chief pigment. If it is not there, B and other carotenoids are present and other C also there, D also there, other things are there, but it cannot prepare the photosynthesis because it will be explained in the next video. Now the thing is, in the chief pigment, one more remember, in the algae you studied already, the chlorophyll, pyophyll, rhodophyll. There in the textbook he has written chlorophyll will have the chlorophyll A and B. Pyophyll will have the chlorophyll A and C. And rhodophyll will have the chlorophyll A and D. What is this? All are having commonly A, as it is the chief pigment. Remember it. Without chlorophyll A, no photosynthesis. That we will discuss later on. Now, chlorophyll A is doing this one. Now, what about the B carotenoids? They are also absorbing. They will send, they will absorb the light and they will transfer the light to the chlorophyll A. Chlorophyll A will receive the light, not only it absorb and it will get the light from the other pigments also, chlorophyll B and carotenoids. So carotenoids and chlorophyll B, they are not directly participating in the synthesis and they are called as accessory pigments. Remember it, this is the important question. They are not participating. Why? How you will prove in this way? So the light is transferred to this one. So it is coming. Light is transferred from the accessory pigments, that like chlorophyll B and carotenoids, to the chlorophyll A, and the photosynthetic rate is more where the chlorophyll A peak absorption is there. There it is showing. This is the important. And the accessory pigments are important for the protection purpose also. What is the protection? Photo-oxidation already told. Means when the oxygen is more, it will spoil the chlorophyll molecules, but it is not happening because of the, it is will be protected by the carotenoids. That much is enough to you. Because xanthophyll will be protect the photo-oxidation, carotenoid will protect the heat, because when the light is falling, heat will generate, that will be protected by the carotenoid. That is out of, not necessary. But the thing is, accessory pigments are important for the protection purpose. Okay. Now, these are the things which I discussed up to now. I hope all you people might have listened properly. And now what my request is, after listening and seeing this lesson, this video, immediately read the textbook. I didn't miss any paragraph also in that one. All the contents I composed, because people will, that is I should not say repeatedly. Now the thing is, so in this one up to now whatever I discussed, so many created questions I told you. If you understand properly this entire thing, concept, you can give any answer to any type of the question within this. Now part 2, and this is the why I done the part 3 parts I done, the photosynthesis chapter. In the part 2 I am going to discuss these sub-chapters. What are they? What is light reaction, the electron transport, splitting of the water, cyclic and non-cyclic photophosphorylation, and chemiosmotic equation, which is more important. This is remember it. That I will explain perfectly. And all these things, like this one it will be. And where the NADP, IPTP and NADP use the primary acceptor carbon dioxide, the Kelvin cycle. and this will come in the part 2. From there to ending, it will come into the part 3. And part 2, it will come, I will upload it within 3 days. So, I think you might, if you receive properly this subject class, please subscribe the channel and post your comment. And if you are having the doubt, my phone number is present on the thumbnail. Contact me, no problem. I will help you. I will give the answers also. And I will support you. That is the thing. Okay?